Good day, everyone. I'm Sadhana, and I'll begin a theme presentation on the future of bioprinting. I will be explaining bioprinting. After me, Sanskriti will cover the biomanufacturing process, and finally, Shriya will conclude with organ-on-a-chip technology. Together, we'll explore how these informations are transforming healthcare. Bioprinting is a type of 3D printing that uses living cells and biomaterials, often referred to as bioinks, to create tissues and organs. Unlike normal printing, where plastics or metals are used, bioprinting prints layer by layer with cells and supportive materials to build biological structures. Next, we'll go with key technologies in bioprinting. Bioprinting can be performed using different methods which use unique benefits. Inkjet bioprinting works like a standard printer but sprays droplets of bioink containing cells. It's simple and precise for small tissue structures. Stereolithography uses ultraviolet light to cure special bioink into solid layers. This helps create very detailed and complex architectures. Laser bioprinting uses laser energy to place cells with very high accuracy while keeping them alive and functional. Applications of bioprinting. Bioprinting has already shown powerful applications in medicine. Regenerative medicine, printing skin for burn patients, cartilage for joint repair, or even components of larger organs. Drug testing, researchers can print human like tissues in the lab and test new medicines directly on them. Personalized implants. By using a patient's own cells, doctors can print tissues or implants that perfectly match, reducing the chance of rejection. The challenges we face in bioprinting. Of course, this field is not without challenges. Material compatibility. Not all biomaterials are safe, and some don't support cell growth well. Scalability. While small tissues can be printed, printing a fully functional heart or liver is still extremely complex. Functionality. A printed organ must not only look like the real one, but also perform all the biological tasks, which is a big scientific challenge. In summary, bioprinting gives users a glimpse of the future, one where organ shortages might be solved, transplants become safer, and treatments are truly personalized. Over to Sanskriti.
Hello, I'm Sanskriti, and I'll be continuing about biomanufacturing, a field that combines biology and technology to produce medicines, vaccines, and even sustainable alternatives to traditional products. Through this presentation, I will explain what biomanufacturing is, the key technologies involved, its applications, the challenges it faces, and finally, its future potential. Starting with innovative biomanufacturing process. Biomanufacturing is the use of living cells and biological systems to produce valuable products such as vaccines, proteins, enzymes, and biofuels. Unlike traditional chemical processes, this approach uses biology itself as a factory. It is central to modern healthcare, drug development, and also plays an important role in promoting eco-friendly production. Next, we'll talk about key technologies in biomanufacturing. There are three major technologies that drive biomanufacturing. First is going to be the fermentation, where microorganisms such as yeast and bacteria produce products like insulin, antibiotics, or biofuels. Second is genetic engineering, where the DNA of organism is modified to create specific proteins. This is widely used in drug development and agriculture. Third is going to be cell culture methods, which involve growing animal or human cells in controlled lab environments. Looking about the applications of biomanufacturing, the applications in biomanufacturing is a vast field. In healthcare, it enables the large scale production of vaccines and therapeutic proteins, as seen during the COVID 19 pandemic. It also supports personalized medicine, where treatments are tailored to an individual patient, making them safer and more effective. Beyond medicine, biomanufacturing supports sustainability by creating biodegradable plastics and biofuels. Next is going to be challenges in biomanufacturing. Biomanufacturing also faces challenges. One is regulatory hurdles. Because these products directly affect human health, they must undergo strict approval process that take time and money. Another challenge is high production costs, which make biomanufactured products more expensive than traditional ones. Finally, scalability, it is a major issue. What works well in small labs is much harder to replicate at an industrial level while maintaining quality and safety. So, exploring the future of biomanufacturing. Looking into the future of biomanufacturing, it is expected to be faster, cheaper, and more precise with the help of synthetic biology, automation, and artificial intelligence. We may soon see engineered organs for transplantation, personalized drug production at hospitals, and large scale sustainable manufacturing replacing fuel based industries. This makes biomanufacturing a cornerstone of both medicine and sustainability in the years to come. To conclude, biomanufacturing is revolutionizing healthcare by enabling life saving vaccines and therapies, advancing personalized medicine, and supporting sustainability with eco friendly products. Next, we'll have Shriya to talk about organ on a chip technology.
So, hello everyone. I'm here to talk about organ on a chip technology. Organ on chip is a microfluidic device which is used to mimic the structure and function of human organs. So, the main core components of organ on a chip device mainly includes microfluidic channels, cell chambers, and membranes. So let's talk about microfluidic channels. These are tiny channels which act like miniature blood vessels, ensuring a continuous supply of nutrients and oxygen to cells while removing waste products. They're also used to stimulate mechanical forces like blood flow or stretching and relaxing the motions of the gut. Cell membrane. So these provide a confined and 3D space for cells to grow and interact. A porous membrane often separates different types of cells mimicking the natural interface with a real organ. Usually, these chips are made from biocompatible polymers like polydimethylsiloxane, which is PDMS. While its popular choice for transparency and flexibility, it can absorb small molecules and affect test results, which is known as a challenge. So, common examples of this organ on chip include lung on a chip, liver on a chip, brain on a chip. So lung on a chip is a model which is designed to mimic alveolar capillary interface of human lung. By applying a vacuum to stretch and relax the membrane, it stimulates the breathing motions. It is mainly used to study respiratory diseases. Liver on a chip is a critical machinery which is used for drug metabolism and used to study drug-induced liver injury and predict the drug toxicity. Brain on a chip are models which are used to study neurological diseases like Alzheimer's and Parkinson's. So next comes the applications of organ on a chip. They are mainly used in toxicity testing. Before testing new drugs in humans, scientists can check how safe they are on organ chips. This helps us to identify harmful effects early and makes drug development faster and safer. The next application being personalized medicine where the chips can be created using a patient's own cells. This way, doctors can test how a particular drug will affect that individual, allowing for truly personalized treatment and strategies. So this also has its own challenges and limitations. However, organ on a chip is still evolving. They also contain some limitations such as model accuracy. It's very difficult to fully mimic the complexity of the real human organs, especially when it comes to multi-organ interactions. The next challenge being integration. Connecting several organ chips together to simulate the entire human body is technically challenging. The last challenge being standardization. For it to be widely used in drug development, the technology must be consistent and reproducible. Thank you.